going to talk to you about the use of AFM cantilevers in a non-conventional way. In particular, I'm going to use them as nanosensors, independent nanosensors, independently from an AFM. So, my institute is the Istituto di Struttura della Materia, that is an interdisciplinary research center in Rome, and our lab is focused on the study of how environmental stimuli can modify can cause a response in living biological systems in terms of morphological, biochemical alterations, and to correlate this with their metabolic response. And to do this, we focus on the use of AFM, coupling it with other kinds of techniques, such as, in particular, spectroscopy, spectroscopy, optical imaging, and STM. The other way around. Okay, so in this presentation I will show you how an AFM can be used in a non-conventional way, and then I'll show you some applications of one of the techniques that we developed, that is the nanomotion sensor. What an AFM is and how the cantilever works, everybody knows it in this room. I'm going to focus on not on the morphological investigations that can be used and can reach very high sensitivity, nor on the nanomechanical investigations, like in this case studying a single bacterium. What I want to do is use an AFM as a movement sensor. So, for instance, if I take an AFM cantilever and I move it on a living biological system, I'm going to have a movement of this living biological system that is transduced by the AFM, that is used at this point as a nanomotion sensor. And so in this way, let's see if it works. Takes some time. Okay. In this way, we can study, the animation doesn't start, but it's not a problem if you want. In any case, we can study clusters of living biological systems, like in this case cardiomyocytes that we obtained through human-induced pluripotential stem cells, and study how the movement of these cardiomyocytes changes over time and exposed to different pressures or different stimuli. By studying this, for instance, we can compare healthy wild-type cells with the diseased myotonic dystrophy cardiomyocytes and determine the form of the beating profile and the frequency of the beating profile using the AFM cantilever not only as a sensor but also as a source of pressure. So we can press more or less on the beating profile and see that while the healthy cells can respond very well and maintain their frequency over time, the myotonic dystrophy cells change their frequency over time depending on the pressure, indicating a different beating tenacity. So, by using this AFM cantilever as a movement sensor, we decided to, probably like this it works better. We decided to combine the movement sensor independently from an AFM and put living biological systems directly on the surface on a cantilever, keeping it detached from the surface of the medium, and we study the movement of the cantilever caused by the fluctuations of the systems on the cantilever itself. For instance, in this case here we have some bacteria that are moving on the cantilever, and we can follow the fluctuations of what we call nanomotion sensor. And we also tagged the same bacteria with a fluorescent tag in order to determine that when we put an antibiotic in the medium, the bacteria stop moving, the nanomotion sensor disappears, and the fluorescence rapidly undergoes photobleaching, indicating the bacterial death. So we determined how to do a nanomotion susceptibility test to study in real time the response of the bacteria to an antibiotic and the antibiotic pressure. And in few minutes, we were able to determine the susceptibility of different kinds of bacteria, gram-negative and gram-positive, motile and non-motile, to different kinds of antibiotics. To do this, also, we performed experiments in parallel to reconstruct an antibiogram of the bacteria to a particular antibiotic and determine with very high sensitivity and in less than 1 hour a complete antibiogram. This is extremely interesting because it's fast and does not depend on the replication rate of the bacteria. The next step was to go to a clinic and pass from the lab to the clinic, and naturally we performed some blind tests directly from positive blood cultures. And what we did was to study many different bacteria, and with a very high success rate, more than 94%, we were able to determine in less than 1 hour the response of the bacteria to different antibiotics. And since this response is independent on the replication rate of the bacteria, we decided to take it a step further and to tackle a problem that is very a medical emergency, both in developing and in developed regions, that is slowly growing bacteria, in particular mycobacteria that are the causes of tuberculosis, for instance, or non-tuberculous bacteria. And also in this case, we noticed how, when exposed to an antibiotic, the response of the bacteria in terms of the motion of the bacterium themselves was very fast, and in less than 1 hour we could measure a reduction in the movement. And this reduction in the movement allowed us to determine, also in this case, different bacteria. This is a tuberculous mycobacterium that is correlated with the cow tuberculosis, while this is a non-tuberculous bacterium, to different antibiotics. We were able to determine an antibiogram also in this case. In less than 2 hours, we were able to reconstruct each of these graphs. And just as a reference, the very same results with conventional techniques require weeks or even months because of the very slow replication rate of these bacteria. Also, the Cantilever and the nanomotion sensor, as a correlation, has the ability to study not only if the bacteria has died, but what happens in between the exposure to the antibiotic and the death of the bacteria. And so we can see how the metabolism of the bacteria responds to the antibiotic pressure. And in this case, with BCG exposed to isoniazid, we could see a periodic explosion in the movement, followed by a stop in the movement, and in a periodic pattern up to the end, to the death of the bacteria, indicating probably that, that the bacteria grow in clumps, and these clumps protect the underlying bacteria that activate after the external layer has died. And so we can not only understand how, if an antibiotic has effect, but also how an antibiotic has an effect. The next step, we studied mammalian cells, and in particular, we used a technique that is typical of single-cell force spectroscopy, in which we went and fished cells with the cantilever. We pulled them out of the petri dish, and we studied the nanomotion caused by the cells, by a small number of cells, or even a single cell on the cantilever. And we performed experiments on many kinds of cells, from mammalian to plant cells, but I'll focus on mammalian cells, and on two neurodegenerative diseases in particular. The first one is the study of how neurons respond to different forms of alpha-synuclein. It is known that alpha-synuclein affects the neurons only if it's in a combined form, in a mixture between monomers and fibrils. And so we studied the nanomotion response of neuron cells in the case of exposure only to monomers, and in the case of the exposure to monomer and fibrils. In the case of monomers, there was no kind of response, even in terms of the nanomotion signal, and we could see very well that after a long period of time, the signal remained more or less constant. In the case of the mixture, it was interesting that at first there was an increase in the movement, which was followed by what we called the popcorn effect on the cantilever. The cells undergone apoptosis, and we had a reduction of the movement. To understand what is happening in this case, we performed experiments coupling the AFM not only with optical microscopy, but also with a fluorescence microscopy. And we exposed these cells and repeated these experiments in presence of Ethinium homodimer 3, that is a fluorescent dye that enters the cell only if the membrane is permeabilized, the cell membrane is permeabilized. And we saw that after very few minutes, the first cells started to internalize the Ethinium homodimer in the case of the mixture, but never in the case of the monomers. So our hypothesis in this case is that the fibrils attack the membrane and permeabilize the membrane of the neurons, and the monomers then can enter to affect the cells and cause the cells to undergo apoptosis. Another experiment was focused on studying Friedreich's ataxia, that is a recessive disease that a genetic disease that causes the cells to be more sensitive to oxidative stress. So, with our collaboration with Cambridge, we were able to study, we were able to develop a new cell line, a model cell, to study how these cells respond in terms of nanomotion, studying what happens when we expose these cells to an oxidative stress. And what we can see here is that when there is no frataxin overexpression, so when the cells are not in some way mimicking the disease, exposed to an oxidative stress, there is a small reduction in the movement, which returns very rapidly to a 100% movement. In the case of mild frataxin overexpression, we have a more complex response, and strong frataxin overexpression causes an even greater response with large moments of reduction of the movement, indicating that indeed the response, the frataxin is the protein that is directly correlated with the response to the oxidative stress. Finally, we tested this nanomotion sensor with many different kinds of systems. In the case of yeasts and fungi, we were able with Candida albicans to reconstruct an antibiogram to a particular antifungal drug. In the case of cancer cells, we studied the response of glioblastoma modified to be susceptible or resistant to a particular drug. In all cases, we were able to determine that there is a direct correlation between movement and life. And in this case, we proposed this, and we are now talking with different space agencies to bring one nanomotion sensor to an icy moon of Jupiter, in particular Europa, or in other systems, to study the presence of life in extreme environments and in faraway planets. Finally, to conclude, I would like to say that we have worked to couple the nanomotion sensor with several other kinds of sensors, that is gene expression, fluorescence, optical microscopy, biochemical sensors, even modifying the cantilever to introduce some nanocontacts to stimulate the cells directly on the cantilever itself. And this coupled with the possibility of working in a chamber in which we can apply physical, chemical, or pharmacological stimuli in order to obtain a device to study the metabolic status of a cell from many different points of view, and to obtain more information on different kinds of metabolic responses of cells. And in this sense, we are very open for collaboration. To conclude, I'd like to thank the group, my group in Rome, and the group from EPFL, the group from Istituto Superiore di Sanità, and the Cambridge University and the MRC in London, in the UK. and thank you all for your attention.